All right. So, you've been collecting molecular data. You've looked at all the different codes, whether it's protein codes or DNA codes, and you look at that sequence of codes and you count up the number of differences. And those numbers of differences give you some data. And how do we use that data to ultimately construct and understand phylogenetic trees? Now, there are different algorithms and different models out there to come up with different hypotheses about the relationship between these things. But I'm just going to take you through a basic way of trying to figure this out based off of some data that you might see. So to start us off here, let's look at, we've got these 5 new species of bacteria, and they're discovered in Antarctic ice core samples. Those species aren't given names in this chart, they're just numbered. The nucleotide base sequences of ribosomal RNA subunits were determined for the new species. The table below shows the number of nucleotide differences between the species. Now, you don't need to know whether it's this nucleotide subunit and all that kind of business. You just need to pay attention to the number of differences. Let me explain how these charts work once. 1 compared with 1 is obviously going to have no differences. When species 1 is compared to species 2, when their sequences are lined up, they found 3 differences. When 1 was lined up and compared to species 3, they found 19 differences. When 1 was compared to species number 4, there were 18 differences in the nucleotides. And when 1 was compared to 5, there were 27 differences. So based off of those numbers of differences, we can tell how closely related species are and how recently they possibly, using molecular clocks and rates of mutations, figure out how long ago they might have been related, but we're not getting there. So ultimately, we have all of these differences. So if I asked you how many differences exist between species 5 and species 3, you should be able to use this chart and say there are 27 differences between 5 and 3. So from this table showing the differences, we can ultimately construct a phylogenetic tree hypothesizing the relationships between these species. And ultimately, I want you to be able to know by the end of this why 5 is where it is, why and 3 and 4 are put together where they are. Okay, so let's start that first. Why is 5 here? Well, when we look at this tree, at this point, this is the last universal common ancestor for all of these different species. Now, 5 branched off from the last universal common ancestor first. And how do we know that? First of all, it's not connected to the other ones. It's branching furthest away. Why is it where it is? Well, when we look at the numbers of differences here, all of these are big numbers. There's not any little ones here. Okay, they're all big, which means that it's not very related to any of these. Okay, so the first thing we do when we look at this is find what we call the out group. We have out groups in cladograms as well, those organisms with the least number of derived characters, but for this one we're looking at which one is most different from all of the others. And then the next thing I asked is why are 3 and 4 placed where they are in the tree, up in this corner? Well, when we look at the chart, which 2 organisms were most recently the same species? So that would be 3 and 4 because they have 1 difference between their nucleotides. So they're not the exact same species. They they no longer interbreed, all that kind of stuff when we talked about speciation, but they have haven't been separated for very long. They haven't accumulated very many differences or substitutions within their codes. So it's taken, they've been split and then they've developed some differences. But this group out here on the very end are those that were most recently a single species. They have the fewest number of differences between them and they're lumped together. And then 1 and 2 here. Now, again, when we look at this, the 3 can be here and the 4 can be here, or the 4 can be on top and the 3 can be on the bottom. That doesn't matter. This is one clade. And then 1 and 2 are also more closely related than they are to 5 or to 4 and 3. When we look at that 1 and 2 here, how many differences do they have? They have 3 differences. They've been separated longer than 3 and 4 have, but 1 and 2 are also going to be more closely related to each other than they are to any of these others. Okay, let's look at another example. So if I'm given a big chart like this, a bunch of names I don't recognize, it's the number of differences. That's what the numbers represent in the chart, the number of differences. And I have this skeleton tree looking thing. Now I'm going to keep on making the trees in a little bit different formats so you can be a little bit more adaptable with this. So the very first thing I need to do for me is I put the numbers on the other side. Sometimes it's difficult for me when it's only half filled out to find that out group. So if I'm looking at it just like this, okay, I know this Africanus and this Ferris are not the out group because they've got a 1 here. Forsteri and Gallus are not the out group because they have a 3. And you can eliminate that way, but here it's easier for me to see this D. polylepis as the out group when I can see the entire column of all of the differences for it. Now, when I look at it this way, you can see 3 out of the 4 differences. And again, why is this one 0? Because it is itself here. There's going to be no differences within that individual species. So, but right here, when I fill this out, you can see now the entire row or the entire column, and you're going to have a preference whether you like looking at rows or columns here. But when they're all the way filled out, now you can see that that has a lot of differences. And it goes on this first branch of the tree because that is where the outgroup goes. That's where the most different one goes. So the next step then would to figure out which 2 were most recently split, and that would go on the other end. And when I look at that, I find the 1, and it's not always going to be 1, right? But the one with the smallest number on the chart is going to give you the 2 most related species, and we put those together where they just recently branched. And then you can see here, this confirms that these 2 are more closely related because they only have 3 differences, but amongst the other species, they have more differences. Okay, another example. Again, another different type of skeleton for the tree. This says the number of differences in the first 60 amino acids of the neuromodulin protein isolated from 5 species. So first thing we do, we need to find the outgroup. And we need to know where the outgroup goes on the tree. Again, I think it's easier if you just fill out the other numbers. And when I do that, this goes 0, 2, 2, 6, 10. So then I just recopy those numbers down here, 0, 2, 2, 6, 10. The gecko is 0, 1, 7, 11, 1, 7, 11. This one is 7, 11. I put 7, 11 here, and then I put 13 there. So that's filled out. Again, now as I look at this, which is most different from all of the others? It's the zebra finch. It doesn't have any low numbers compared to the other ones. So then the next thing I do is I try to find the smallest number in the chart, and those are going to be the ones that are most recently species separated, and that would be the turtle and the gecko. So then I need to figure out what goes in those last two spots, and now it's different the way they're branched right now, because it's not just finding, okay, what's the next biggest number, because when I look at the next biggest number here, I still have these, and this also leads me into this, but let's talk about that for a second. Okay, so the two that we don't have are finch and frog, right? So when I look at finch and frog, these aren't necessarily going to be the same numbers here, okay? And I'm picking one. I'm not just putting two in, because it's not finding these that are closer, that are linked together, because the finch is more linked into these guys than the finch is linked into this turtle one, or the frog one. So you can see here, if we have turtle and gecko as the most closely related, and I know I have to put finch and frog in here, which one is going to go closer to the turtle and the gecko? The finch, because it only has two differences with each of those, whereas the frog has 7 differences, and it'll be further away. So then finch goes here, frog goes there, and then we're done. Okay, last one. Which species is the outgroup and least related to all of the other species because it has the most differences from all of the others? And then where does it go on the phylogenetic tree? Panda would go there, because panda has all big numbers in this chart. It has the most differences from everybody else. Okay. Next, which two species shared sorry, I got the answers in here. Which two species shared a most recent common ancestor and have diverged most recently because they have the fewest differences? Where do they belong? Um, Again, even though I messed up on my slideshow here, you can see the black and the brown have one, and they go there. Okay, and then polar goes in the last spot, because that's where it goes. And that's how we use this data, this molecular data, to try and understand and make hypotheses about phylogenetic trees and the relatedness of species. So hopefully that's helpful.